Good evening, everyone. Madam Winnie here, Biochemistry Simplified. I thank you so much for the subscriptions, for the views, for the comments, for the shares. Let us work together, let us walk together, and let us learn together so that we can achieve our goal of simplifying the understanding of biochemistry as a subject and as a course. I'm done with the introduction to biomolecules. I've introduced much on the carbohydrates, on the lipids, on the proteins, on the nucleic acids, the much which the students need to understand as fundamentals of biochemistry. So I'm diving into metabolism. I've already done much of the carbohydrate metabolism, where we dived into glycolysis, which takes place at the point when the glucose level is high. So we break down our D-glucose to be specific, substrate specificity or optical specificity of enzymes. So we have to break down our D-glucose following the 10 steps to pyruvate, with the main objective of generating energy in form of ATP. And at the point glycolysis takes place, insulin as a hormone regulates the pathway. Then we looked at during starvation. The glucose level is low. The body is looking for the sources where to get glucose. It has to synthesize glucose from the non-carbohydrate precursors, and that is the pathway which we discussed, known as gluconeogenesis. So the non-carbohydrate precursors which we mentioned were amino acids, glycerol, and lactate. And at that point when gluconeogenesis takes place, glucagon as a hormone regulates the pathway. So it means there are bypassed steps in gluconeogenesis. The steps in glycolysis, which are irreversible, they are bypassed in gluconeogenesis. If you refer to the previous section of the carbohydrate metabolism, we'll understand much about that. Then we dived into the TCA cycle, because under aerobic condition, the pyruvate, which is generated from the glycolytic pathway, is converted to acetyl-CoA. So we are talking about the amphibolic role of glycolysis. It always provides precursors for other pathways. So the acetyl-CoA, which come from the glycolytic pathway, will be converted to acetyl-CoA via pyruvate dehydrogenase complex of enzymes, and then the acetyl-CoA goes to the TCA cycle for generation of reducing equivalences FADH2 and NADH, which act as electron carriers in electron transport chain and oxidative phosphorylation. Then we looked at the pentose phosphate pathway. From the name, it generates the pentose sugar, which forms the structure of the DNA and RNA. If you look at the structure of the nucleotide, as I mentioned in my nucleic acids introduction, the nucleotides being the building block of the nucleic acids, a nucleotide will have a phosphate, a base, and a pentose sugar. So the pentose sugar, which forms part of the nucleotide, is generated by the pentose phosphate pathway. Pentose phosphate pathway also generates NADPH, which is used in lipid metabolism. Fatty acid synthesis is using NADPH. Integrity of the red blood cells needs NADPH. Synthesis of the hormones, the estrogens, the steroid hormones, it needs NADPH, which is generated from the pentose phosphate pathway. The other product generated from that pathway is ATP because it acts as a shunt. In the non-oxidative phase, we generate the glyceraldehyde 3-phosphate, we generate the fructose 6-phosphate, which can go to the glycolytic pathway to generate energy in form of ATP. So I've covered those pathways in carbohydrate metabolism, but today I would like to dive into amino acids, meaning proteins metabolism. So we are looking at amino acid degradation. So when you talk about metabolism, as I mentioned in carbohydrate metabolism, metabolism involves digestion followed by absorption, then we have degradation and synthesis, and lastly excretion. So even at the point we are looking at the protein metabolism as a whole, we don't dive much into digestion and absorption because that bit a student can understand, can read and understand. The bit which becomes difficult, the structures, the enzymes, how important the pathway is the bit we are covering. So after the digestion, the proteins, the simple product of digestion will give us amino acid. Remember in class, the classification of proteins based on their composition. We have simple proteins, which the hydrolysis will give amino acid. And then we have the conjugated proteins, which their hydrolysis will give an amino acid. And based on the component which is present there, it could be a phosphoprotein, it could be a metalloprotein, it could be glycoprotein. So metalloprotein, mostly they have the iron 2 present in them, and then we have the glycoprotein, which has a component of a carbohydrate on it. Phosphoprotein has a component of phosphate on it. Nucleoprotein has a component of nucleic acid on it. So those are complex proteins. But for the simple proteins, the hydrolysis, normally when protein is hydrolyzed by the peptidases or the proteases, they are going to give us amino acids. So today we are going to dive into amino acids degradation. And if you look at amino acid degradation, by remembering the structure of amino acid, we have the amino terminal end, we have the carboxyl terminal end, we have the alpha carbon, we have the hydrogen up there, and the R side chain, which gives the physical and chemical properties of those amino acids. So based on the R side chain, we classify our amino acids into 7 classes: the aliphatic, the alcohol containing, the aromatic, the basic, the acidic, the amide of acidic, the sulfur containing, based on the R side chain. So we also classified them based on the nutritional value. We talk about the essential and the non-essential. Then we classify them based on the number of carboxyl and amino group. So we're talking about the monoamino, monocarboxy, diamino, dicarboxy, monoamino, dicarboxy. So today we are going to look at, based on the product of the degradation of amino acids, they will be classified as glucogenic. The degradation product is pyruvate, and any other intermediates of the TCA cycle. The TCA cycle is the Kreb cycle. It is also the citric acid cycle. So if the degradation products of that amino acid give us pyruvate, or oxaloacetate, or alpha Kg, or succinyl-CoA, or succinate, or fumarate, it is glucogenic. But sometimes when amino acid is degraded, or it is broken down, the degradation product can be a ketone body. We are going to see it in lipid metabolism. The ketone bodies are beta-hydroxybutyrate, the acetone, and acetoacetate. If it gives any of those three, the beta-hydroxybutyrate, the acetoacetate, and acetone, which are ketone bodies, then it is known fully as ketogenic. But sometimes, some amino acid degradation products will give us one of the glucogenic and ketogenic. So it can give us any of the ketone bodies and intermediates of the TCA cycle or pyruvate. So they are also classified in the third class as both ketogenic and glucogenic. So by definition, degradation will yield pyruvate or one of the intermediates of citric acid cycle, then it's known as glucogenic. Or the other definition of glucogenic amino acid is they can be converted into gluconeogenesis, they can be converted into glucose through gluconeogenesis. So through the gluconeogenic pathway, already oxaloacetate is in there, pyruvate is in there. So it's just the same definition as the first one, but the first one is easy. The product of degradation is pyruvate and any other intermediate of the TCA cycle. So examples of glucogenic are glycine, serine, cysteine, methionine, glutamate, aspartate, asparagine, glutamine, threonine, proline, histidine, alanine, valine, and arginine. Then ketogenic, purely ketogenic, we have lysine and leucine. If you think about the lysine was falling under the basic, and then leucine was falling under aliphatic, the subclass branch chain amino acid. Then the ones which give us both glucogenic and ketogenic are tryptophan, phenylalanine, tyrosine, and isoleucine. This slide summarizes the degradation products of the amino acid. So there the key shows the glucogenic ones are the ones in red, ketogenic are the ones in green, and then both glucogenic and ketogenic are shown in purple. Somebody can say that one is pink, but based on how we see the color, those are the key to what we are seeing as the degradation products of those amino acid. For example, let us look at asparagine and aspartate. You know, asparagine is an amine of aspartate. Just by adding an amine group on R side chain, you will get asparagine. So you can break down asparagine to aspartate via the asparaginase, and then after aspartate it forms oxaloacetate. Oxaloacetate is an intermediate of the TCA cycle. Remember at the time the acetyl-CoA combined with oxaloacetate to form citrate, so that brings clearly that it is glucogenic. In other words, still oxaloacetate through gluconeogenesis can be used in the synthesis of glucose during starvation, because we define gluconeogenesis as synthesis of glucose from non-carbohydrate precursors, and amino acids are one of the non-carbohydrate precursors, and in this case asparagine will be broken down to aspartate. Aspartate will be converted to oxaloacetate. Oxaloacetate can go to gluconeogenesis to generate glucose during starvation, or it can enter the TCA cycle for generation of reducing equivalences FADH2 and NADH. The other one is isoleucine, which was branch chain amino acid. It can be converted to acetyl-CoA. The acetyl-CoA can enter the TCA cycle by combining with oxaloacetate to form citrate, or it can go to form acetoacetate, which are ketone bodies. So in this case, the isoleucine is both ketogenic and glucogenic. Alanine, cysteine, glycine, serine, threonine are converted to pyruvate. The pyruvate can be converted to the acetyl-CoA. It goes to the TCA cycle. Or the pyruvate can also go to gluconeogenesis. So they are known as glucogenic amino acid. Phenylalanine, tyrosine, tryptophan. Remember, they were both ketogenic and glucogenic. The degradation products will have an intermediate of the TCA cycle as well as a ketone body. So they are going to form acetoacetate as well as having an intermediate of the TCA cycle. So they are both glucogenic and ketogenic. Leucine and lysine are purely ketogenic. Then we have arginine, proline, histidine, glutamine. They are glucogenic. Glutamine is amide of glutamate. The amide of glutamate is glutamine. Glutaminase, which will fall under the class of hydrolases, presence of water, will remove the amine group from glutamine. It will form glutamate. Glutamate will be converted to alpha-ketoglutarate, which is an intermediate of the TCA cycle. So they are defined as glucogenic amino acids. Then you have valine, threonine, and methionine are specifically going to form succinyl-CoA, which will further form succinate in the TCA cycle. So those are intermediates of the TCA cycle. They are purely glucogenic. That is valine, threonine, and methionine. Isoleucine is both glucogenic and ketogenic. So classification of those amino acids based on their degradation products, they'll be classified as glucogenic, ketogenic, as well as both glutogenic. Let us look at a few of the examples on the degradation of those amino acids. Asparagine, you know, it has an amine group here, an amino group as well, carboxyl group, hydrogen, alpha carbon. So if you look at asparagine, we were classifying it as diamino dicarboxylic. So asparaginase is a hydrolase. You see water is coming in, and then we remove the amine group which was existing on the R side chain. The NH2 is removed here. So when you remove it, you remain with aspartate. Then the alpha Kg to glutamate, they act much as amino carriers. They carry the amine group of the aspartate. So we have the aminotransferase, which will convert it to oxaloacetate. So the oxaloacetate is an intermediate of the TCA cycle. So that one makes it glucogenic. Glutamine converted to glutamate, glutaminase being the enzyme, still addition of water, removal of NH3, you'll have glutamate. Glutamine was still dicarboxy, diamino, based on the number of carboxyl group, but glutamate now is dicarboxy monoamino. So we have glutamate. Glutamate will further be converted to alpha ketoglutarate, the enzyme being glutamate dehydrogenase. The alpha ketoglutarate is an intermediate of the TCA cycle, so they are glucogenic. Then you look at histidine here. Proline also is going to be converted to alpha Kg. Arginine is going to be converted to alpha Kg, though I didn't stress much on those pathways. I just looked at a few. Histidine is also going to be converted much into alpha Kg, and that one makes it glucogenic. But then there is an importance of tetrahydrofolate here. The folic acid, vitamin B9, will be helpful so much in most of this pathway. So they have to be present at the point when the enzyme responsible for the metabolic pathway is working on such a step. So tetrahydrofolate should be present there, but then at last histidine is converted to alpha Kg, and those are glucogenic amino acids. The amino acids which are converted to pyruvate, you can see alanine, just one step. Alpha Kg and glutamate still, they're carrying the amine group which is on the amino terminal end of those amino acids. But look at the specificity of enzymes, optical specificity. We defined optical specificity as the enzymes involved in carbohydrate metabolism will only work on the D sugars, not L. Then the ones for amino acid metabolism will only work on the L form, not D. So you can see how the optical specificity is represented here. So the L-alanine is converted to pyruvate. That one shows glucogenic amino acid, the alanine aminotransferase catalyzing the one step. Glycine has to be converted to serine, then serine further converted to pyruvate. Still, the importance of vitamin B9, the tetrahydrofolate, the folic acid has to be present here. Glycine can further be converted to carbon dioxide and ammonia, but then serine hydroxymethyl transferase will catalyze the conversion of glycine to serine, and then serine is alcohol containing. The presence of the OH on the R side chain, if you look at our notes on the classification based on the R side chain for those amino acids, so the dehydration, it will remove the OH which is present there, as well as the amino group which is on the serine will be removed. So we'll have the formation of pyruvate. So we classify glycine, serine, and alanine as glucogenic. Cysteine is also converted to pyruvate. There are several reactions, but at last we have pyruvate. And then you can have threonine, which is converted into pyruvate and further into succinyl-CoA. So those are examples of glucogenic amino acid. A bit of the branch chain amino acid, the valine and isoleucine, will be further converted to succinyl-CoA, which is in the TCA cycle. Succinyl-CoA was that step where now the succinyl-CoA has to be converted to succinate. So succinyl-CoA is an intermediate of the TCA cycle. That one makes valine and isoleucine, which fall under branch chain amino acids, or the major group aliphatic, being glucogenic because of their degradation product being succinyl-CoA, an intermediate of the TCA cycle. So they are glucogenic. The ones which are both ketogenic and glucogenic, I didn't explain further the steps, but then the last product of their degradation is fumarate, which is intermediate of the TCA cycle, and acetoacetate, which is a ketone body. So that one makes phenylalanine and tyrosine to fall in both ketogenic and glucogenic amino acids. A bit of the metabolic disorders. You know, by the time a metabolic pathway takes place, it involves much of the enzymes. And what comes clear is the specificity of enzymes, how specific they are to their substrate, how specific they are to their reactions, how specific they are to their groups, the types of bond, if it's peptide bond, if it's glycosidic bond, if it's an ester bond. So at that point where there is a defect in that enzyme, there is a problem. That is what relates to the metabolic disorders. And in most cases, they will show as a disease or a disorder. So we are talking about amino acid metabolic disorders. Just to mention a few, we can look at the phenylketonuria. Phenylketonuria, the keto, the urea, urine, the keto group, you know, the ketones. So it is caused by absence or deficiency phenylalanine hydroxylase. You know, if you look at the structure of phenylalanine, it is aromatic, yes. There is a CH2, then the aromatic ring without the OH. If you add the OH down there, it's tyrosine. So converting phenylalanine to tyrosine is just by adding the OH. That enzyme catalyzing that step, conversion of phenylalanine to tyrosine, is the one which is defective here. So the one which is known as phenylalanine hydroxylase. So in this case, if it is defective, the phenylalanine is not converted to tyrosine. Rather, it is transaminated to phenylpyruvate and phenylactate. And that is what leads to ketones in urine as well as in blood plasma. There is high level of that. So there is high level of phenylalanine because it can't be converted to tyrosine. And now there is formation of phenylpyruvate. You know, if you look at the structure of pyruvate, it is COO minus, but then the second carbon is C double bond O, and then CH3 down here. It's 3 carbons. So the presence of that keto, the second carbon, if you look at phenyl lactate, so there is high levels of phenylalanine because it has not been converted to tyrosine. There is transamination, formation of phenyl pyruvate and phenyl lactate in the tissues, plasma, as well as urine. So I talk about phenylketonuria. If you do such tests in the lab, you will detect the high levels of ketones. And these ones come handy in clinical biochemistry. That's how systematic they go. So you start with the biomolecules, you are introducing the amino acids. You go to metabolism, the degradation products of these biomolecules or their building blocks. Then at the point now where there is a disorder, the clinical biochemistry will come in because the patients will present themselves that they're not feeling well. So a test has to be carried out in the lab. We are testing for a chemical component, and you have to request for the correct sample. In this case, we are talking about the urine and even the blood plasma. You'll request for the urine sample. The patients have to go for urine sample. Whatever you're going to test there is the ketones present in the urine. So in clinical biochemistry, which comes after metabolism, is there is always the normal level for all these in the system. The level of the ketones, the level of the sugars, the level of glucose. But there is a level when it is higher. It is more than the normal level. That is where the problem is. So the patients have to present themselves in the hospital, because at first we talked about buffers. So the physiological pH has been tampered with. There is a problem. So in that case, you have to test for what is higher than the normal level, and you have to ask for the correct sample. So in this case, for the phenylketonuria, the phenylalanine hydroxylase is defective. Phenylalanine cannot be converted to tyrosine. There is accumulation of phenyl lactate, phenyl pyruvate, as well as the phenylalanine, because it can't be converted to tyrosine. So those are a bit of the symptoms: CNS, mental retardation, failure to walk or talk, seizures, hyperactivity, tremor. The brain weight is below normal, and this lead to life expectancy being shortened. If you look at genetics, the phenylketonuria falls under the autosomal recessive disorders. We'll be able to touch on that when the right time comes. For now, we have to understand at what point which of those enzymes were defective and lead to which disorder. So the hydroxy, and it leads to transamination of phenylalanine to phenyl pyruvate, phenyl lactate, and that is what is found in the urine and blood plasma. The other one is albinism, genetically determined by lack of tyrosinase. Look at where the tyrosinase works, where the melanin is forming. We are still on the aromatic degradation of aromatic amino acids, phenylalanine and tyrosine to be specific. That point where you can see it converts dopa to melanin, tyrosinase is defective. This is the pathway where the catecholamines are generated. You can see the norepinephrine and epinephrines. They are generated by this pathway, aromatic amino acids. So tyrosinase in melanocytes oxidizes tyrosine to dopa and dopashinone. But when that one is defective, no generation of melanin. Albinism will come handy. So inhibition of production or lack of melanin in skin, hair, and eyes. And these individuals have high sensitivity to sunlight. Alkaptonuria. We are still talking about disorders relating to aromatic amino acid, phenylalanine and tyrosine. Still that one pathway. Since the phenyl hydroxylase is defective, it's the one which controls the whole pathway, then everything will go wrong. alkaptonuria will come handy here. Absence of homogentisate oxidase. You can look at this block. Formation of mallyl acetoacetic acid is affected. So alkaptonuria, you'll know it much, production of that homogentisate oxidase. If it affects the child, a diaper, when exposed to air for some time, will turn black. For adults, we have some of the characteristics which we are going to discuss. So the urine will turn black when exposure to air. You can see the one in black, that is for the alkaptonuria, but this is for the normal urine. So the homogentistic acid is accumulated and excreted in urine, that is alkaptonuria. So a bit in children, the urine in diaper may darken. In adults, darkening of the ear, which is showing. The eye, the sclera and cornea of the eye, there are dark spots. There are dark pigments deposited in the intervertebral disc of the vertebrae when you do such tests. And treatment is, you should give low phenylalanine and tyrosine in diet, and that will reduce the level of homogentistic acid. The maple syrup relates with the branch-chain amino acids. So it leads to accumulation of these branch-chain amino acids, and the enzyme which is defective here is branch-chain dehydrogenase. So a bit of the characteristic is the patient is dehydrated, vomiting, patient has feeding problems, and there is severe acidosis. Due to the odor also of the urine has a smell of maple. This can also lead to mental and physical retardation. So the main treatment is you have to limit the amount of the